So hi everyone, in the last video we have seen completely about cephalosporin, and today we are going to see about aminoglycosides. We will see the structures, most important points, its mechanism of action, and along with that what are the important points asked in the different competitive exam and semester exam. We will cover all these things in today's video, and the video will be completed within 15 to 20 minutes. Okay, so let's start. First of all, aminoglycoside. See, these aminoglycosides are very important antibiotic, and these mainly acts on aerobic gram negative bacteria. This mainly acts on aerobic gram negative bacteria. First thing. Second thing, in aminoglycoside structure, you will see already it is written in the name only. Amino means amine group will be present. Glyco means glycosidic bond will be present, and last me in glycoside, that means in this glycosidic structure will be present, glycose sugar will be present, and along with that amine group will be present. You will see guanidine structures also present in that. Now I will draw the structure and I will tell you. Okay, and what are the drugs present in that? The most important drugs are streptomycin, kanamycin, neomycin, tobramycin, gentamicin and all. Okay. So streptomycin. In streptomycin, streptidine structure will be present. So that's why it is called streptomycin. Then neomycin. In neomycin, you will see a 5-membered ring will be present. Not ring, sorry, 5-member structure will be. Since 5-member structure will be present, that is quaternary. That's why neo word came. Neo. Due to the presence of 5 carbon. Streptomycin, neomycin, then kanamycin, then tobramycin. This tobramycin is very importantly used, and then gentamicin. These are most important drugs, comes under the class of aminoglycoside. Now what is the mechanism of action? It is very simple. You have to understand. For example, the beta-lactam rings or beta-lactam antibiotics acts by inhibiting the cellular synthesis. How it inhibits the cellular synthesis? They binds to the penicillin binding protein present on the cellule of the bacteria. In the same way, it inhibits the protein synthesis of the bacteria. In that case, cellular synthesis inhibits. So ultimately what will happen? The body fluid gets inside the bacteria and the bacteria dies due to bursting or collapsing. And here what will happen? In the mechanism of action of aminoglycoside, you have to remember, aminoglycosides inhibit the cell, not protein synthesis, not cellular synthesis, protein synthesis. So when protein synthesis is inhibited, ultimately functional protein will not form, functional enzyme will not form. Ultimately what will happen? Metabolism will be disrupted inside the bacteria, and due to which ultimately bacteria will die. That's it. Now, how it disrupts, how it inhibits the protein synthesis. So, during protein synthesis, you got everything in central dogma: replication, translation, transcription. So, see, DNA to DNA replication, DNA to RNA transcription, and RNA to protein translation, right? So, in that case, what will happen? From RNA to protein translation, in the case of translation, you will see the mRNA will come here and it will bind here. And when the mRNA will come and bind here, what will happen? This, what is this? 50S and 30S. What is that? Ribosome. So it will come here and it will bind to the ribosome. And in that case, what will happen? Translation will take place. So in that, mRNA will come and reading of the codes and codon will take place. After that, it will synthesize the amino acid. Who will synthesize? That ribosome will synthesize the amino acid, and it will go through the exit slit. That amino acid will go, and then what will happen? Protein will form. This protein will act in the form of enzyme, right? And then biosynthesis and everything will take place, metabolism will take place, and the normal process will go on. But here what will happen? When this mRNA will come, the misreading of mRNA will take place. Why the misreading of mRNA will take place? These aminoglycoside drugs, these drugs will come and it will bind irreversibly to this 30S ribosomal subunit. In this 30S, it will bind to 16S rRNA. Okay, this is very important point you have to remember. this 30S ribosome, in that again 16S rRNA, in that it will bind. So when it will bind irreversibly, what will happen? When this mRNA will come, it will block here and it will fill the space. At that time, what will happen? During the reading of codon for synthesis of amino acid, what will happen? It will start misreading that, and when misreading of amino acid will take misreading of RNA, that information will takes place. In that case, that amino acid which will be formed, it will be non-functional. So when it will be non-functional, ultimately whatever the protein will form, it will also be non-functional. Okay. So in that case, what will happen? That protein will disrupt the metabolism of the bacteria, and ultimately bacteria will die. Okay. Now we will see the structure. So this is simply the mechanism of action. So how the aminoglycoside will work? It inhibits the protein synthesis, and when protein synthesis is inhibited, metabolism disrupts. When metabolism disrupts, bacteria will die. What are the side effect or unwanted effect of this amino acid? These are very important. So simply you remember, amino acid is a mean guy. It will slap on your ear. So when it will slap on your ear, what will happen? You will have, what will happen? Ototoxicity. Ototoxicity. And it will kick on your kidney. So when it will kick on your kidney, what will happen? Nephrotoxicity. Nephrotoxicity. And it will leave you paralyzed. So when paralyzed means neurotoxicity. These are very important. Neurotoxicity. These are most important side effect or unwanted effect of aminoglycoside. Now, if a woman has taken aminoglycoside and she is pregnant, and if this aminoglycoside kicks on her kidney, what will happen? Again, what will happen? Teratogenicity will happen. Ultimately, the fetus will be defected. Yes or no? So it will also cause teratogenicity. So you have to remember these 4 points. Very important. Okay. Now we will see the structure, chemical structure of amino acid, and this is also not amino acid, so aminoglycoside, and this is also very simple. For example, take streptomycin. So in that case, this is the central ring, this is the glycosidic bond, and in this glycosidic bond you will see amino sugar are present. What are these? Amino sugar. In that you will see a aldehyde group present here and an alcohol group present here. Here you will see an alkyl group present. These are very important. Here you will see one guanidine group. So what is guanidine group? Guanidine side chain. What is guanidine side chain? When a single carbon is connected, with 3 nitrogen atom, and in that 1 nitrogen atom is connected with double bond and remaining 2 nitrogen atom is connected with single bond, that is called guanidine. So can you please tell me, this aminoglycoside is guanidine derivative or not? Yes, it is also guanidine derivative. Okay, so another guanidine is present here. So 2 guanidine present and remaining are alcohol. So one OH, another OH, here you will see another OH. Okay, here you will see one NHCH3. This is very important, and here you will see one CH2OH. These two are very important, this CH2OH and NHCH3. Okay, why? Because it will ultimately show the effect. Okay, so this is the structure of streptomycin. What is this? This is the structure of streptomycin. Okay, and what is this? Streptidine. After that, what is that? This 5-membered structure, L-streptose. And this one is N-methyl L-glucosamine. So this one is glucosamine. Glucosamine means in this, see, what is that? Ether group is present, yes or no? So this is glucosamine. That means amino sugar. Gluco means sugar and samine means amino group also present. So that is amino sugar. that is also amino sugar. And what is that? 6-membered structure. Okay. So this is very important. Now in that SAR, you will see these two rings will be considered as same. Sorry, these two, because both of them are amino sugar, and this is 6-membered structure. Okay. So now in that, the most important point is this CHO. The CHO CHO is essential for the activity of aminoglycoside. Here, if this OH is replaced with hydrogen or any alkyl group, then there is no effect. Okay. In that again, this CH2OH, that is alcohol group. Presence of this alcohol group is very important. So it is essential. Again, this NCH3 group, it is also already I have mentioned earlier. It is essential for the activity. These are necessary. Presence of this ether bond are must necessary. This is also called glycosidic bond. These are must and necessary. Must and necessary. So up to this you have to remember these are very important things. Okay. Now why it is called streptomycin? Due to the presence of that streptidine. In the same way, in the case of neomycin, you will see 5 carbons are present, and that is quaternary. Quaternary means like this. So 1, 2, 3, 4, and another is present in the center. So this is called quaternary, and in that 5 carbon will be present. This condition is called neo during the common naming. That's why neomycin is called neomycin. In that neo came from this structure. Okay. And see, in the case of streptomycin, due to the presence of this streptidine ring, it is called streptomycin. Okay. So these are all about aminoglycosides. Now please repeat after me, or just remember, revise again from first to last. First of all, aminoglycoside acts mainly on aerobic gram-negative bacteria. Okay. Second point, aminoglycoside acts mainly, what is the mechanism of action? By inhibiting the protein synthesis. Yes, and binding to the 30S ribosomal subunit. Again, in the 30S, it will bind to the 16S rRNA. Okay, how it will inhibit the protein synthesis? By inhibiting the amino acid synthesis. No, by misreading of the mRNA. Okay. Next, what are the side effect or unwanted effect? First of all, aminoglycoside is a mean guy. It is a mean guy, so it will slap on your ear. When it will slap on your ear, ototoxicity. Second, it will kick on your kidney. When kick on your kidney, nephrotoxicity. Third, it will keep you paralyzed. So when it will keep you paralyzed means neurotoxicity. Number 4, if a pregnant woman is taking the aminoglycoside, and if the aminoglycoside kicks on the kidney, what will happen? Teratogenicity will happen. That fetus will be defected, yes or no? So that's why teratogenicity also unwanted. This is the trick to remember. Okay. So, and most important thing, this SAR is not so much important, but the structure of aminoglycosides like streptomycin, kanamycin, neomycin, tobramycin, gentamicin, structure of all these are very important. In the next video, I'll explain all the simple structures individually. Okay, or else I'll upload individual structures video in short. Okay, if you see this video is supporting to clear your concepts, then please do subscribe and like. Thank you.